program for us here. She has lots of experience. I'll let you introduce your journey and what you've
Oh,
done here.
sure,
But we're
sure.
grateful that she battled traffic to get in here today to be a part of the speaker
It's
series. So thank you.
Thanks, Dr. Malecki. It's great to actually, I welcome coming in here and seeing all the faces of my colleagues and the students. Usually when I come in, my office is on the 8th floor and no one's there in the hallway. And then I come to the 9th floor and all the doors are closed. And I go, why did I come into work? But I have tried to 10. Okay, well, I will try that. So, okay. So yeah, the only time I've seen people is when I've made a previous lunch plan and then met up with them when I get in here. As Dr. Malecki said, I am a professor in epidemiology. I come from a laboratory background. I worked at Rush for a number of years in gynecologic oncology research, and I got interested in epidemiology and data analysis, and I decided to do my master's work here at UIC. I got an MPH in Epi, and then I started the doctoral program while I was working here as a project manager, and finished my doctorate. Did a postdoc here. You can see I'm pretty ingrained in the Chicago area, and have been faculty since then. And so I have worked with many people for many years here. And some of the studies that I'm going to tell you about today, um, particularly the Great Lakes fish consumption study, I've been working on that since before I started the doctoral program. So it's been something of a real long-term and really productive project. Anyway, so this is the outline for today. Um, and I'm going to really focus on the first two studies the most. But then I wanted to mention the fish study I did with Dr. Buchanan and some NHANES studies, because I think that might be of interest to students in EOHS too. That NHANES is such a rich data set for many purposes. And I'm not really focusing on our new faculty. There's other things going on in Epi Bio that I'm not focusing on, but these are the projects I'm mainly involved with. Okay, so let's see. Um, okay, there we go. Okay, so Great Lakes fish consumption study. I put all my funding up here on all the slides when I'm talking about a study. Important to make sure that you always put funding on your presentations. And so this study why are we focusing on Great Lakes fish? Well, there's the Great Lakes is particularly Lake Michigan and Lake Ontario and Erie and Huron really have a history of a lot of industry around the Great Lakes. And so they have become polluted. And the pollution has become a problem, especially in the sports fish, at the top of the food chain. Um, it's a fishing does have an important role for many populations who live around the Great Lakes, for anglers, for Native Americans, and there's some commercial fishing on the Great Lakes, but it's pretty minor. Um, so there's a significant amount of contamination in these Great Lakes fish, and including mercury, but mercury's in a lot of fish, commercial fish and sports caught fish, but particularly Great Lakes fish have higher levels of some of the persistent organic pollutants. They also they have beneficial nutrients. So we want to really make sure when we tell people about risks of Great Lakes fish consumption, we also consider beneficial nutrients. All right, so for those who don't know, I'll just mention what persistent organic pollutants are. So these are things, these are chemicals that persist in the environment for long periods of time. Many of them are highly lipophilic, and so they accumulate in organisms in the water and then ultimately concentrate up the food chain. Not all of the there are a few that concentrate up the food chain that aren't stored in lipid tissues. Mercury is one of those that's stored in muscle tissue. PFAS chemicals also are not lipophilic, but they also bioaccumulate in fish. So, well, I mentioned there's a lot of consumption of sport caught Great Lakes fish, and this project originated. The ATSDR funded a number of studies back in the early 90s, some of them laboratory research and some of those population studies to understand the health risks of Great Lakes fish consumption. And the study I'm telling you about today was initiated by Andy Anderson up at the Wisconsin Department of Public Health. He formed a consortium of all the Great Lakes states to develop this study. And they used state charter boat, Great Lakes charter boat license lists to recruit participants for the study. And then this tells you the time frame that this study took place in. They used random digit dialing to recruit people who had low levels of Great Lakes fish consumption. Of course, you can't use random digit dialing anymore because no one's area code matches where they live at this point. So we have been following a group of these participants since then, and we've collected extensive information on fish consumption, other dietary factors, and health history. And we've measured exposures across 3 different time points. And you can see, so PCBs, polychlorinated biphenyls, DDE is a metabolite of the organochlorine pesticide DDT. PBDEs, the polybrominated diphenyl ethers. And both PCBs and PBDEs have been used as flame retardants in many products across the years. Recently, we've added measurements of PFAS and a panel of metal and metalloids to the rich exposure history that we have. Okay. And then we've also collected a lot of markers of physiologic state of the participants, including serum lipids, inflammatory markers, steroid and thyroid hormones, markers related to glucose metabolism, and autoantibodies. And so the main outcomes we've studied are diabetes, thyroid disease, cardiovascular disease, and we did find some impact of the fish consumption and POPs exposure on uterine fibroids in some of the participants, as well as strong signal for diabetes, especially from that metabolite of DDT, and lots of associations with thyroid and steroid hormones with PCBs, that actually we've seen much stronger relationships in the women in the cohort than the men in the cohort, which is interesting. So so this is an endocrine disruption study, right? And in this field, there's a lot of work that doesn't necessarily distinguish between gender. And it's really important, though, to do that in this work. And so we've really focused on gender specific effects. And the PBDEs were weakly associated with Great Lakes fish consumption. The PCBs are really strongly correlated with Great Lakes fish consumption. And when you compare the Great Lakes fish consumers to data from the NHANES, the National Health and Nutrition Examination Survey, you see that they have substantially higher exposures. Susan.
Do you know specific dumping incidents of PCBs into the Great Lakes? I mean, is that the main source of just industrial dumping?
I think it's industrial pollution. I believe there's a number of Superfund sites along the Great Lakes. Different lakes have different mixtures of chemicals too. I don't know if any of you remember Dr. An Lee. She did a lot of work across the years looking at the levels of many chemicals in the sediments of the Great Lakes, and they're not equally distributed across the lake. There's hot spots around, so I think it's dumping. I think it's pollution. Just in the general region. Remember, there's many tributaries that flow into the Great Lakes that can also bring pollution. So anyway, these are some of the past studies we've done. I've just have some data from a couple of dissertations that EPI students have done with this data. This one is Mike Blakovich did this a few years ago. He looked at coronary heart disease. We had mainly focused more on the diabetes and thyroid disease, and he focused on coronary heart disease. And he showed a risk of these are hazard ratios. And so the hazard ratio for coronary heart disease is 1.6 for a doubling of serum PCB levels. So that's his main model, but I thought I would present these other models because I think they're very interesting. So the second model he looked at, he added the base model is controlled for a lot of confounding factors that you would expect, age and sex and BMI. But he added hypertension and diabetes separately to the models. Can anyone think of why he might have added them separately to the model? Anyone who's not an epidemiology student? Because you've all had some epidemiology. What would we be worried about that he might have not wanted to put those two factors in the main model? Not confounding, but something, you're on the right track. Well, I've already told you we've seen some associations of diabetes with these persistent pollutants, right? So if you have diabetes associated with the pollutant, and you've got cardiovascular coronary heart disease associated with the pollutant, what might happen if you put diabetes in the model? It could be a mediator, right? So we want to make sure we don't mask the effect of the exposures on coronary heart disease. So we're careful about putting other diseases that might be mediators of that effect. Because remember, hypertension and diabetes are risk factors for coronary heart disease. So I think this is a good example of kind of the thought patterns of epidemiologists and how we work through this. So in this case, he really didn't see much change in the effect of PCBs on coronary heart disease when he added hypertension or diabetes to the model. So that suggests it's not really a confounder or a mediator. And so what we would conclude as epidemiologists is we don't need to control for it, right? So this first model tells us what the risk level is. I hope I'm not getting too far in the weeds here, but it's interesting. I want to give you an idea of what we think about when we're looking at this data. Okay, so the final model is a model without lipids. So we always control our analyses of PCBs for lipids, because remember I told you they were lipophilic chemicals. So the lipid levels in a person's body can impart some bias into the measurements. So we always control for that, right? If you have more circulating cholesterol and triglycerides in your body, you have high lipid levels, you may have more POPs circulating too, because they circulate associated with the lipid levels. So we always control for lipids. But we do it in different ways. I won't go into that. But in this last model, Mike partially controlled for lipids for the bias in lipid measurement, but not for circulating lipids. And there you can see that that attenuated the effect estimate. It went from 1.6 to 1.36. So what did that suggest? Epidemiologists can answer this question too. Megan, what do you think it suggested? No, there's no effect modification in this. Yeah, it's a confounder. It's important to control for it because it's a confounder, or potentially a mediator. We can't just tell from looking at the model what it is. We have to have a conceptual model to help us figure out what it is. All right. I think I've pushed this slide far enough. Let's see. It's 12:20. Okay. So I may skip a few slides, but I did want to show this because I think this is really interesting. And this also from a student dissertation. We use mortality data. So what this is actually, this project was funded by the one of the ERC pilot grants to the student who was doing the work. And she needed the money because it costs money to get data from the National Death Index. And we wanted to link our cohort through their things like names and location and age to National Death Index data and see if fish consumption and POPs exposure affected mortality. And so this first table on the left shows the results for total fish consumption. So this is fish meals from the Great Lakes, commercial fish meals, inland lake fish meals, just anything that they ate. And you can see, so these are These are incident rate ratios, I believe. And so these are, red is the lowest level of fish consumption, orange is moderate level of fish consumption, and yellow is high level of fish consumption. So you can see that the risk of cardiovascular disease mortality decreases as overall fish consumption increases. Does anyone have any thoughts on why that might be? Well, Omega, it's something that would be a good hypothesis, that there's beneficial nutrients in fish that are protective for cardiovascular disease. In fact, there's a lot of data in the literature that suggests that's true. So we do see that in this cohort. When we stratify by the second group are those people who had low levels of fish consumption, of Great Lakes fish consumption. And these are the captains who were eating more Great Lakes fish. When we stratify by those two groups, we see a stronger effect of fish consumption on people who don't eat Great Lakes fish than on people who eat Great Lakes fish. So then Dr. Liu did the same analysis looking at low, moderate, and high levels of Great Lakes fish consumption. She could only do this in the captains because they were the ones who were eating Great Lakes fish. And what she saw, this is not a significant association. So basically this association that we saw for total fish consumption is not there when we look at Great Lakes fish consumption. So that suggests that there's something in the Great Lakes fish. I'm sorry. It's possible. I mean, and we don't have that data really. I mean, we didn't do standard nutritional surveys that you would need to actually look at that hypothesis. So this study took place. I think we went through 2015. So one of our plans for one of our students is to update the mortality data. Because of course, as the cohort ages, we're going to find many more cases of diabetes and coronary heart disease and cancer. And so we're going to, we feel like we saw some potential borderline associations with things like cancer and diabetes. And so we think we may see more. And we have more exposures to look at now than we did when this study was done. So in this way, we're going to extend this analysis to a new expanded analysis with an OEE student.
I have a question about the Great Lakes fish. Is the classification of Great Lakes fish versus other fish like the type of fish, or there unique fish in the Great Lakes? Like, is that
so it's really, we ask specific questions about where they obtain the fish, and that's how we did that classification. The Great Lakes are actually stocked with a lot of sport fish that aren't naturally found there. And so lake trout is a native species, but the rainbow salmon and things like that are not, and they're actually stocked into the lake for recreational fishers.
these are people who knew where their fish were.
They knew where their fish were coming from, and in fact, we have information about the specific lake they came from, and we've been looking at that. Megan, who's doing this great study, has been looking at the new PFAS measures we have in Great Lakes fish and the amount of fish people consume from the Great Lakes and then overall. And we see number of the PFAS chemicals are related to Great Lakes fish consumption. And there's some relationship with commercial fish, but it's a lot weaker. So that suggests that this might be a really important source of exposure. And there's other data across the country that suggests that sport fish is a big important source of PFAS exposure. Yeah, so. I mean, so it's really exciting, and all these samples were back from 2015, so it's not new samples, but it's new measurements that we have, and we can answer questions at least about the Great Lakes fish back in 2015. So the other thing, well, I don't know if I have time to talk about this. I'm just going to mention this really briefly. And if anyone's interested in this for some of their other work, you can contact me and I can talk to you more about it. But one thing that we're really interested in EpiBio is chemical mixtures, because there's a variety of factors that people are exposed to. And this is really important for occupational health too, because the people who get specific exposures through occupational routes also have this vast background exposure to many, many chemicals. So, this is a huge topic in environmental epidemiology. Dr. Julia Bauer, who's joined us recently, has done a lot of work with mixtures too, related to metal exposures mostly. And so we had a grant from NIH to actually help develop methods. And we have biostatisticians in EpiBio who have worked with a lot of these methods and can be part of student dissertations or students' master's work if they're interested in thinking about chemical mixtures. So I don't think I have time to go into this anymore, but that would be the main people we've worked with on this would be Dr. Chen, Wayun Chen, Saria Awadala, and Sanjeev Basu, who are all really great biostatisticians. And we collaborate with them all the time on these really important issues. So I do think this is an area that occupational epi could build up expertise in and incorporate into some of their studies and student work. I don't know how everyone else feels like that. The EOHS faculty feel like that, but I mean, you can't. The traditional way where we look at one exposure at a time just doesn't tell us the whole picture. And you may have one occupational exposure that's really strong. Maybe that's enough to just look at that exposure, but it might be good to start thinking about all the other exposures people get through their day-to-day life. So we're happy to collaborate on things like that. I'm not going to spend time in that. I'm going to talk next about the SOUL study. It's called the Study of Latinos Hispanic Community Health Study. And this is a multi-center national study. One of the sites is in Chicago. And across the last more than a dozen years, they recruited Hispanics from different sites across the nation. And it's not just Mexicans, it's Mexican, Puerto Rican, Cubans, Dominicans, Central and South Americans. So it's a really diverse population of Hispanics. And the focus of the study is cardiovascular, cardiometabolic disease in Hispanic populations. And as you might guess, there's very rich acculturation data available from these study participants. And this is a longitudinal study, so I think they completed a 6-year follow-up, and I think they're just completing the next 6-year follow-up. So it's a really strong study design. Martha DeViglis over in the Center for Minority Health Research at the Medical Center is the PI here. And this actually is useful for occupational data too. One of our OEE students is looking at shift work and its effect on kidney function and inflammation in the study of Latinos. So this is really an important research at UIC that certainly students in EOHS could tap to use in dissertation work too. Being a very large multi-center study, there are a number of things you have to do to go through to get the data. But we've been doing this for years. There's paper proposals you have to write to get permission to get the data. And we can put you on IRBs and things. So if anyone wants to have some ideas about using this data, please use EpiBio as a resource to facilitate getting the data. This is just some background. I'm not going to read through this slide. This is just some background about the population. But you can see that the baseline exam, there were over 16,000 participants across these 4 study sites. So it's a very large cohort. And so we have what we call an ancillary study. So they allow ancillary studies to be proposed by investigators to add on different types of measurements. It could be survey measurements, it could be measurements using stored samples. And that's what we did. We accessed the stored samples from the baseline, and we measured a panel of those persistent pollutants that I told you about before, as well as hormones and inflammatory markers. And so this is diabetes, hormones, and POP study. And a lot of our students have been using this data and are currently using this data for their dissertations. And it's not the whole 16,000, it's a subgroup of the 16,000. And we selected people who had a baseline visit and who came back for the second visit at 6 years. And we took half of the people have normal blood glucose at baseline and half have prediabetes. And the underlying goal is to understand the whole the effect of hormones and chemicals on stages of diabetes transition. So people they're not just normal glycemic one day and all of a sudden the next day they have frank diabetes. It's a long transition pathway. And so that's what we've been looking at. But it's over 2,000 people with these POPs measurements. So let's just skip those. So we have, you can use these measurements, right? It's a cohort study. So you have the exposures measured at baseline, and then you have multiple outcomes measured across time. And so you can look at other outcomes with the exposures. So we've had a number of students work on different outcomes, including kidney disease, coronary heart disease, obesity, inflammation, fatty liver, hearing loss, and then there's also genetic data on a good 80% of the participants. And so You can look at interactions between genetic risk for certain diseases and the POPs exposures to see if there's effect modification and interaction. The people who have genetic risk for disease and higher exposure, if they're at essentially greater risk than you would expect of developing the disease. So some of these are, I mean, these are all in some of them have been presented as dissertations, and some of them are just being started. I think we have paper proposals approved for almost all of them. And so this is an enormous amount of work, and we're going to learn a lot about these outcomes and POPs exposure. And, you know, the Hispanic population is really important to study for some of these outcomes because particularly diabetes and fatty liver, they have substantially higher prevalence than some of the other ethnicities in the US population. So these are really, some of these outcomes are particularly important to study in a diverse Hispanic population. Actually, while I'm on this, if people are interested in things like cardiometabolic outcomes, one of our students who is not in the OEE program because she's an MPH PhD student, so she has funding from that program, but she's going to be defending her dissertation in about 2 weeks. I think it's 2 weeks from today. And she studied metal exposure and diabetes and hypertension in a Mexican American population from Texas. So if this kind of, if you're intrigued by this kind of research, try and come to that dissertation presentation, and it will be announced. I think it's, is that one? And so it's right. So it's 2 Wednesdays from today at 1 o'clock. So I encourage you to go to these dissertations if you're interested. And okay. So I'm going to skip that data because I'm running a little late. The other thing I wanted to mention, and I know Dr. Buchanan's presented in the community cafe about the fish intervention study for health. So I'm not gonna go into details about that, but I did want to mention that we do have ongoing work, and we do have students working on some of the data from this project. Right now I have an MPH epi student who's working on it. And we've also have a pilot grant to look at PFAS exposure in some of the fish from Chicago markets, and then to measure PFAS exposure, to add that measurement to the mercury assessment that we did in this study, because that's becoming, as I mentioned before, fish consumption may be an important route of exposure to those chemicals. So if anyone's interested in this study, talk to Dr. Buchanan or myself about what data might be available. And then the last thing I wanted to mention is NHANES. Everybody knows what NHANES is. We've been exploiting NHANES data because it's publicly available. It doesn't require IRB approval to use the data. And it's an enormous data set. It's clean data. It's really collected in a standardized, reproducible manner. And what's important to us in environmental epi is that they have been assessing biomarkers of environmental exposures for at least 20 years. Actually, I think it goes back further than that because they were looking at lead even before they started adding more exposures to their measurements. If this is something that interests you, my EPID 536 class in the spring, actually, you will learn how to use this data set and then develop your own analysis from the data set, which is really great experience. You do need to have some epidemiology this after the intro to epi though. So you would need EPID 404 or some equivalent to be able to take the class and get the most out of the class. But some of the students, not every student, but a few students have gone on to actually publish their data analyses. And so I was looking over my CV in preparation for this talk to see how many environmental exposure related NHANES publications we've done. Some of these students were OEE students. There's Julia J. Anglin is Julia Bauer, so this was during year MS that she did here. So she worked on a paper with Dr. Buchanan and myself. So we looked at methyl mercury there. We've done other papers on lead and cadmium exposure and sex hormones. Stephanie DeFlorio Barker, who was a OEE student in EOHS, did a paper with me on phthalate metabolites and bone mineral density. Several OEE students looked at arsenic and obesity. Several others looked at PFAS and fish consumption. And then we have another dissertation paper from Yang Liu on methylmercury intake in Asian and non-Asian populations. There's more. Jessica Madrigal, another trainee, looked at cadmium and kidney function. This is not an EOH, this is a PhD student, but not an EOHS trainee, looked at arsenic and lung function. We looked at PCBs, dioxins, and furans. This was part of an OEE trainee dissertation. I just met Jessica again, looking at OC pesticides and sex steroid hormones. Kizu Kim looking at metals and metal mixtures and thyroid hormones, and Chibazar looking at mercury and lipids. So you can see there's just a ton of data here on all kinds of outcomes and all kinds of exposures. And so, and there's occupational data too, that could even be combined with some of the exposure data potentially. I don't know enough about the occupational data that's there to know how refined the data is, but certainly. So if you're interested and really want to learn epi analyses as part of your training, take EPID 404 and then take the class, and you'll learn how to use this data. And then it might be part of your dissertation or your thesis work. So